Good afternoon, everybody. Thank you for coming. We have an opportunity to celebrate success today, which is always fun to do. Just about a month ago, we had our ESD award ceremony, which was a lot of fun as well. So I always enjoy it when we get to do this. And today we bring BSD and ESD together to celebrate 2 DOE Early Career Awards that have been awarded to a staff member in each of the 2 divisions, Melanie Mayes and Wellington Muchero. It's very exciting. These are really huge career boosts. These awards are very competitive. They come with $500,000 a year of research money for 5 years, so $2.5 million total award. Really, really hard to get. Every year we have several people who apply for them in every division, and year after year we come back empty-handed, even in spite of excellent efforts. But this year we landed 2 of these within the directorate, the ESD directorate. So we're very proud of both Melanie and Wellington, and we're going to have them tell us about their projects, what they're planning to do, and then we'll have some refreshments afterwards and mix and mingle. So Melanie's going to speak first, and I'll introduce her, and then Wellington will speak after, and Tony Palumbo will introduce Wellington. So Melanie has been with us here at the lab as staff since 2002. She's in the Environmental Sciences Division, where she is a team leader for the what's the name of your team, Melanie? Multi-scale processes, biogeochemical. And she was made a team leader about a year ago, correct? Yeah. And so that was along with the reorganization of the what's now the Earth Sciences group that Eric Pierce leads, and she's doing a fantastic job there. And she holds also 2 joint faculty appointments at the University of Tennessee. One in the Biosystems Engineering and Soil Science Department, and another in the Earth and Planetary Sciences Department. And that is the department where she actually got her PhD at UT, and in geology. And she also is an avid hiker. She loves the outdoors and nature, and that has spilled over, in fact, into her extracurricular activities out in the world. She serves as the board chair of Way South, which is a nonprofit organization promoting environmentally and fiscally responsible transportation projects in Appalachia, and I did not know that until I got this bio from her today. So this speaks to Melanie's broader impact on the community, and because she has consistently been out in the community contributing, she also received a Knoxville YWCA honor in science and technology in 2015. She was part of the NG Tropics Significant Event Award last year, which was for the successful contribution that ORNL made to the Biological Environmental Science Research Office, their new project on next generation ecosystem experiments in the tropics, and Melanie was a key part of that. And in 2014, she was inducted as a fellow into the AAAS, American Association for the Advancement of Science, which is a high honor, high career level honor. Prior to that, she had won the Stanley Auerbach Award here in the Environmental Sciences Division. Going back to 2011 also, she won the Young Alumna Award for Professional Promise from her alma mater in the Earth and Planetary Sciences Department at the University of Tennessee. And that's only about half of the awards and honors that are listed on her CV, but I'm going to stop there and turn it over to her so that she can tell us about her early career award proposal or project that she's planning.
Okay. Is that all right? Yeah, that's good. Okay, perfect. So I'm going to do this again, which is thank you all for coming. I really love seeing all of you here, and really I'd like to thank all of you. Some of you know, like, I was a baby when I came here, so I really, really appreciate the time I've had here. So I'm going to talk about a comprehensive framework for modeling emissions from tropical soils and wetlands. And I would like to thank from the deepest heart of my heart the Biological and Environmental Research Program at DOE for giving me this award and giving me some awesome things to do for 5 years. So it's about the soil carbon cycle. And so several years ago, we wrote an LDRD proposal. And what we did is we did other things, but in general, we built this microbial enzyme decomposition model. And what we wanted to do was do a better job at modeling releases of carbon from soils. And what we wanted to do specifically was to be more mechanistic about this. And so we built it with explicit microbes, explicit enzymes. So microbes send out the enzymes. The enzyme goes into the organic matter and litter. It makes a really specific chemical reaction. And it depolymerizes some of that carbon and makes dissolved organic carbon. And this is what the microbes actually take up. And they emit CO2 as waste. And so this is essentially what the MEND model looks like. It was really successful. We managed to hire Gongxing Wang. Sindhu Jagadama is a new faculty member in the Biosystems Engineering and Soil Science Department at UT. And this was, in fact, converted over to ORNL's TES SFA, which is led by Paul Hansen and Dan Rasciuto. So this has been great for us, and we've learned a lot about soil carbon cycling. But something that we completely have neglected that people have asked me about since we built it is, are you considering redox processes? The soil carbon cycle is, in fact, a redox couple. And so what that means is, this is organic matter. Organic matter gets oxidized. It makes CO2. Here's the valence change, negative carbon to positive carbon. At the same time, there's another half of this, which is oxygen. Oxygen is what we call a terminal electron acceptor, and it gets reduced all at the same time. Aerobic decomposition is really well represented in most models. They may or may not include microbes. Our model does. But we're still missing a whole lot of what's going on, which is, in particular, something that happens in wet soils. So this proposal was about tropical soils, and it's about wetlands, okay? So what happens in wet soils is that products can build up, especially in the tropics. There are these dense, clayey soils. They get a lot of rain, a lot of sudden rain. And so what happens is that all this CO2 and other products can build up, and oxygen can, in fact, be consumed. After that, what we get is methane production from these products right here, okay? But methane production is not as easy to simulate as that aerobic decomposition that I said is generally well covered by models. This is a model intercomparison project, WetChimp. This was published in 2013. And they basically said there are two things wrong with how we're simulating methanogenesis. There's an incorrect structural representation of the carbon cycle, and there's inadequate identification of wetlands. And so my project addresses both of these things. So what we really need is a comprehensive framework to transition between those aerobic soil decomposition processes and anaerobic decomposition processes. So again, this cycle here with oxygen as a terminal electron acceptor, and this cycle here with CO2 and methane. So when we lose oxygen, when we get really wet soils, we tend to fall down here in this anaerobic regime. But something else that I haven't mentioned yet, which is nitrous oxide, is another important greenhouse gas associated with this. And so what happens in this case, once all that oxygen is gone, the first thing that happens is microbes start reaching for other things to get their energy from. In this case, nitrate. And that's how we ultimately get production of N2O. By the way, for those of you that don't know, both methane and N2O are also very important greenhouse gases in addition to methane, or in addition to CO2, which is what we typically think about. Both of these guys tend to be a little more powerful. They have a greater global warming potential, although they may not have quite the same residence times as CO2. They're not as long. So this is important. But in fact, there's more to it than that. There's an entire redox ladder. So this is sort of in the lexicon of geochemists who like to think about all of these redox-sensitive processes. So basically, everything on this side are various terminal electron acceptors. And so after you run out of nitrate, then you start going into manganese and iron and so on. So if we want to comprehensively know how we get from here to here, we might also have to consider these, depending on where you are. And we know from studies from one tropical soil that iron reduction can account for a significant amount of carbon fluxes from a soil, up to 40% in this case. We don't know as much about tropical soils as temperate soils. And so this is pretty limited data. That's another reason why we're focusing on the tropics. The other thing to think about is that this reaction can also release phosphorus. This is a very important limiting nutrient for tropical environments. So this is why we're doing it, basically. And so here I'm going to talk a little bit about what I'm going to do and where I'm going to do it. In Puerto Rico, which I have been working under the NG Tropics framework for about a year now, there's a series of basically valley and ridge transitions. So Puerto Rico goes from sea level all the way up to over 1,000 meters. And it's very, very wet. They get lots of precipitation in the mountains there. And in addition, there's this very small but intense sort of micro-topography. So what you're looking at here is this very, very wet valley soil. Just right up there at the top of the diagram is a ridge. So we know about this particular location that these valley soils especially can produce significant amounts of methane, whereas up on the ridge you may get more CO2. So I'm going to work, there are a number of sites in Puerto Rico that are well studied, but not in quite this sense, and that are accessible. So this is the first place I'm going to do research. And what I'm going to do, you may have noticed this little guy in those last photographs. I have a field scale instrument for measuring greenhouse gases. It's a Picaro G2508. It's a cavity ring down spectrometer, and it can measure N2O, methane, and CO2. And it has 12 flux chambers associated with it. And so basically these little guys just open and close as a function of time. As long as you feed it electricity, or in this case gas, it can run continuously, get continuous long-term data. So of all of these different gases. So this is really exciting. And you can also get the flux chambers far enough away to where we can do a valley and ridge transition at this site. So this is the first thing we're going to do. And here is actually, I hooked this up in Puerto Rico last month, in fact. And so here is literally the first data to come off of this thing. Of course, it's no good. But anyway, we were very excited to have it going. And we'll also take measurements of soil moisture, temperature, oxygen content, and soil water chemistry to get a really complete understanding of what's going on in the field. And then, after we are satisfied with Puerto Rico, we are going to go to Panama. And this is the Changuinola peat deposit. This is a world-famous San-Ponsac wetland. It's very large, and it's very well preserved. It's really quite a treasure. I have not been there yet, but I'm very excited to go. And so we're going to work at this wetland. Here's a picture of it. The other cool thing about it actually is there's a phosphorus gradient across the wetland. So this should be really exciting. So we'll have 2 sites, for the most part, that we're going to work at. Now, that's going to tell us about what's going on in the field, but it may not tell us as much as we might want to know about mechanisms. And that is the value of lab scale data. So I have another really cool instrument. This is a micro oxymax respirometer. And this, just like the field scale instrument, is capable of getting continuous data, continuous reading for CO2, O2, methane, H2S, and hydrogen as a function of time. And it has the capability to have 20 different flasks hooked up, so we can get a lot of data out of this thing. And so what we can do then is We can use an incubator. We can vary soil moisture. We can vary oxygen content. We can vary temperature. And we can add, in lab scale manipulations, a lot of these different terminal electron acceptors to see how the system responds. And this is what the data will look like. This is micrograms of CO2 as a function of time. So we could compare, say, control and treatments. And in fact, this one was a phosphorus addition experiment. So we've been doing some of this work under the LDRD that we have right now, which I'll talk more about in a minute. So that's going to tell us about how the geochemistry will affect CO2 respiration. But of course, one of the things we want to know is why. What microbes are there and what are they doing? And so what we will do is we'll use metagenomics, metaproteomics, metatranscriptomics, and a variety of other microbiological techniques to look for the genes, to try to understand the microbiology of these soils, and to try to understand them better to be able to predict it. And so this is something, there's lots and lots of data about genes. So this is someone else's publication. And the point I want to make here is that if you want to know about this process from acetate to acetyl phosphate, basically, we sort of already know what those pathways are. So all you have to do is do your analyses and then look for these genes to control production of these enzymes and all of those different enzymes. So we can go into the system knowing exactly what we're looking for and go find it in the genes. So an example of that comes from our current LDRD project. And so some of my collaborators are here in the audience, and I owe these guys a great deal of thanks for starting this kind of work so that I can talk about it without being stupid. So what we're doing in this case is we went to the Gigante fertilization experiment. This is in Panama also. This is a long-term field manipulation. They have been adding all of these different nutrients to this place since 1998. So this is long enough to where you would expect that the microbial community has actually changed in response to its environment. And so we went there a couple of years ago and we collected control sites and phosphorus addition sites. And we just went in and got a couple each because we wanted to just do a pilot study and see how it went. And we did all the DNA extractions. We sent the data to the JGI, and they sent us back with a lot of information. And so what we focused on, our LDRD is focused on phosphorus acquisition. And so what we did was we looked for genes that were in greater abundance in the control plots versus the phosphorus acquisition plot, phosphorus fertilization plots. And so all of these enzymes in particular were found in much greater abundance in those control plots than the phosphorus addition plots. All of these enzymes are about phosphorus acquisition. So this tells us that this long-term experiment has indeed affected the microbial community and that we can detect it. So we can exactly do a transfer of all this knowledge into my early career. So I'm very excited about this. And also into the next NG Tropics. So I will here give a really big thanks and a plug for ORNL's LDRD program. You've now heard that 2 parts of my early career came through LDRD first. So that's a great resource here. Okay, so what we're going to do then, from all of what I was just telling you, we're going to end up with this wonderful detailed genomic and geochemical model. We're going to understand a whole lot about these processes. We're going to know who's there and what they're doing and all the greenhouse gas emissions, how it's affected by oxygen and soil moisture content. This is awesome. But what this does not do is allow you to go beyond, say, something the size of this room to a region or to a globe or something like that. So we have to figure out a way to take this really detailed information and to simplify it so it can be applied to bigger scales. And that's really where the nut is in this field right now. We can probably do some of this stuff, and people are doing it to various degrees, but we don't know how to scale it up. So that's a big thing that's going to happen in this project. And I don't know how we're going to do it, but we will figure it out. So we have to simplify. We have to take something really complicated and detailed, throw it in probably some kind of statistical magic kind of thing, and figure out how we simplify whole lots of detailed information into just a few microbial functional groups, okay? These are just conceptual. I have no idea if this is the answer, but that's what we're going to do. And we're going to do this in the MEND model. The MEND model will be a starting point, and it will help build men to go from an exclusively aerobic model to one that meets that transition between aerobic and anaerobic. And then, finally, the other point that Melton made about simulating methane had to do with wetland inventory. So this is a wetland inventory around the globe. And this, of course, is very coarse scale. There are probably better ones than this, but this was nice to show. It's what I went and got off the web. But at any rate, we are going to build a better wetland inventory so that then we can apply all of what we've learned and figure out how important it is at different sites. And so this basically is the full conceptual model. Detailed geochemistry, simplification, upscaling. And this is the endpoint for the project. And so in conclusion, I want to really put some specific thanks out here to all of these folks. My husband, Gray, is in the audience. He puts up with me. He takes care of dogs when I travel. There will be more of that. And all of these folks here have been really integral, I think, to the success of this early career. Some of these folks were co-PIs on the first LDRD. Some of these folks are associated with the second one. Some of these other folks gave me some great review comments and helped me build this thing. And so, and again, I would like to thank ESD and BSD. You all have been so awesome to me, as well as the Climate Change Science Institute. They have helped really build this synthesis between experiments and models, and that's what BER wants. That's what this project is. It's part of the reason why it was funded. So the CCSI has been a great resource, and it's a great thing for us here. So, thanks. Oh yeah, so they're taping me, and that's why they're going to insist that you use
the microphone.
And
why Kim is running
DOE is going to see this presentation potentially, so okay. Thanks DOE. This rocks.
Yeah. In your sort of simplified model when you're moving forward, when I saw you sort of had the fungi, the lithotropes, the bacteria, are you really thinking just the bacteria just as the anaerobes? And is that because you think the fungi are the most important aerobes?
I think in tropical environments, the fungi tend to be in the litter layer, and they tend to get a lot of nutrients from just the litter layer. So a lot of trees don't root extensively down into the soil in the tropics, or if they do, they're just getting water, they're not getting nutrients. So I have some reason for thinking it might be like that, but don't read too much into that thing either.
So Melanie, when you do your lab incubations, are you going to characterize the microbial community kind of across the gradient of phosphorus in Panama, and then the ridges and the slopes, kind of when you're doing Puerto Rico work, or where your soil's coming from, and kind of what are you going to do with that? Definitely in Puerto Rico, we'll be looking at valley and ridge transitions. The Panama site, actually, I haven't been there. The lead author's name is Alex Cheeseman, and he has a study where they were looking at phosphorus gradient across it. And so I think that's probably a really attractive thing, is to hit that same gradient, but I haven't been there, so I'm not totally sure. But it seems likely it's where the LDRD would take us, so yeah. Very cool.
Melanie, cool stuff. I think it's in the magic you described as simplifying the model, but I wonder if and as you incorporate microbial genomic information into the model, and they are in some way regulating what the model is doing, have you thought about what that does to data requirements as you go to extrapolate that to the globe? what sort of thought have you given to how well we might be able to characterize the microbial community in a global model?
That's it. So the scaling thing is really the tough nut. What's your level of what to lump and what to split? And I do not have an answer. Clearly, there are going to be data requirements. So this, I was talking about the value of doing this intense data collection. I mean, you can't use that in a global model, right? Although, having said that, though, many of them function at half-hour time steps. So I don't know. That's definitely going to be an issue. What I hope kind of falls out of this is that wetland inventory in our work maybe helps us decide how to lump and split the wetlands. And then maybe you have to go out and do more investigations on this kind of wetland and this kind of process. So my two sites are very different. One of them is an ombotrophic peat bog and the other one is soils. They're going to be very different, and that is by no means going to answer that question. Hopefully the wetland inventory will give us a start on where to go next, where to do more of those kind of specific kind of studies. Does that answer your question?
Yeah, I mean, yes.
Okay.
It's going to be hard.
Yes. I've got 5 years. Anyone else? Gary.
Congratulations.
Thanks, Gary.
That's very nice. So you've had an interesting journey to get to this point. And
Yeah.
so my question won't be technical, but as you look back and reflect how you started your career in certain areas of geoscience and where you've ended up now, what words of wisdom would you give to some of the other early career folks that are here and seeing how you were able to reach this level of success?
Okay. Let's see. Be open and be flexible. So for those of you who don't know, my background is in geology and contaminant transport. My master's and PhD thesis were on the transport of radionuclides in soils. There were no microbes in any of my system. And if there were, I totally ignored them. They might as well have not existed for me. And it wasn't about carbon, and it wasn't about really any of this stuff. It wasn't about climate. But somebody asked me if I could help with something in the carbon realm. Hey, can you help with this field study? I'm like, it's how stuff moves through dirt, no problem, I can do it. And I got very interested. So that's the thing, keep your mind open. If somebody asks you to help with something, well, maybe go and help them, because it might take you in a direction you didn't anticipate to go. And when I started doing that, I also sort of started thinking about what was important. And I thought, well, radionuclides and toxic metals are very important at many places, but at the same time, this climate is this big monkey on our back that's going to be very important. And so I said, well, this may be a really good place to take my research. So be open. I think the other thing is to look for the holes, look around the organization where you are and say, what's everybody doing? Is there something in here I can do that no one else is doing? And Gary, actually, you and I had a conversation that really helped me crystallize it. And I don't remember if you remember exactly what you said, but it really made me think about how I would fit into the space and what I needed to do. The really cool thing about ORNL is that you can have so much flexibility into doing anything that you want. We're a mission-driven agency, and so if the need is there and you have the initiative and the interest, you can go after it. It might take 3 or 4 tries on a proposal, but by 3 or 4 tries, you should get it. You'll have a better proposal by then, and somebody will believe that you know enough and they'll give you the money. But, don't give up. Keep trying. Thank you all.